This experiment was performed to extract DNA from mammal blood following the Mohammed and Seyviban protocol from 2009, modified by Dr. Victor Rodriguez. When we talk about the big picture of DNA extraction, it has three main steps: the lysis of the cell, the proteinization that involves the separation of the phases, and the recovery of purified DNA. Nonetheless, when we want to do DNA extraction from animal cells, the procedure will be slightly different from DNA extraction from plant cells. This is due because of the cell's structure. One of the main differences between them is the cell wall that is present in plant cells. This wall has different compounds that make DNA extraction more difficult than in animal cells. In animal cells, instead of dealing with metabolites, we deal with a range of proteins, lipids, white blood cells, red blood cells, platelets, and plasma, which can contaminate my DNA sample. In this laboratory investigation, we attempt to extract DNA from mammal blood donated by one of our classmates so we can compare both methods.
We first took 2 tubes and labeled them. Then we add 100 microliters of blood in each tube and 100 microliters of ACT solution. We proceeded to centrifuge at 250 Gs for 10 minutes. After this time, we removed the supernatant. We proceeded to add 500 microliters of extraction buffer and 5 microliters of proteinase K. After this, we incubated the solutions at 65 Celsius degrees for 30 minutes. Right after the incubation, we put the tubes into ice and add 500 microliters of chloroform isoamylic acid. Now we centrifuge at 11,600 revolutions per minute for 10 minutes. Once again, we removed the supernatant and we repeated the 3 previous steps again.
After removing the supernatant, we added the same volume of isopropanol and 150 microliters of potassium acetate, and we left them in the freezer overnight for the DNA to precipitate. After this, we centrifuged our DNA at 13,000 rpm for 10 minutes and 4 degrees Celsius. After this, we proceeded to remove the supernatant and dry the pellet at 37 degrees Celsius. We then proceeded to perform our washes, first with 700 microliters of absolute ethanol mixed by inversion. After this, we centrifuge at 16,000 rpm for 10 minutes. We removed the supernatant and continued with our second wash. This time we added 100 microliters of 0.5 molar ammonium acetate plus 500 microliters of 70% ethanol. We performed another centrifugation, this time at 13,000 rpm for 5 minutes. After the 5 minutes had passed, we removed the supernatant and dried the pellet for 30 minutes at 37 degrees Celsius. After our tubes were dried, we resuspended our DNA in 50 microliters of TE buffer.
The TE buffer consists of trees and EDTA. We use it to elute DNA and to keep and store the DNA in order to use it in other experiments. EDTA protects the DNA from degradative enzymes called DNases. EDTA binds divalent cations that are necessary for DNase activity. Trees is the main buffering component, and its chief role is to maintain the pH of the buffer at a stable point. The function of the protein case, as the word says it, is to digest the proteins. The SDS detergent breaks apart the lipid membrane and solubilizes cellular proteins. Ethanol helps to remove the remaining salts and SDS from the preparation. Adding a salt such as sodium chloride to a solution containing DNA neutralizes the negative charges of the DNA molecule, making the separated DNA molecules more likely to collect together and become visible. The DNA must be separated from the other cellular components using the phenylchloroform isomyl alcohol, PCIA reagent.
Blood samples are of extreme value to the field of genetic research, especially to forensic genetic laboratories that have to take genetic profiles from blood that can be deposited on a wide range of supports. The method developed by Mohammadi and Seyviban is a simple, reliable, and quick procedure for the extraction of DNA proceeding from mammalian cells. It also provides a yield of resulting DNA that's comparable to previous developed methods. A study carried out in 2013 in which DNA was extracted from buccal swabs, hair samples, urine, and blood samples, demonstrated that there is a possibility for low levels of contaminants in the DNA isolated from the serum or plasma that are much less abundant rather than those that exist in urine and buccal swab specimens. In this same study, after the PCR procedure with the extracted DNA, hair, and blood PCR products produced excellent isolated digestive products, as the ones that we can see in the PCR running showed in the video. Besides the commercial kits available in the market, there is a great need for efficient, quick, and cost-saving methods for animal DNA extraction. The standard protocols for DNA extraction are still time-consuming, frequently needing expensive enzymes, and most of the materials that are used routinely are toxic. It's also necessary to conduct an efficient DNA extraction procedure that does not undergo extra and unnecessary steps that risk the process of further contamination or DNA degradation. In an attempt to attain these goals and simplify the procedure, a study conducted by Sarah Samadhi Shams and colleagues describe a less hazardous, simpler, inexpensive, and rapid protocol for extracting high-quality DNA from blood samples. The initial lysing and precipitation was conducted with a red blood cells lysing buffer, RLB, consisting of 3-hydrochloride buffer, with the difference being that the first precipitation was carried out using magnesium chloride, used because it binds to DNA and thus protects it against DNases, enzymes that are in direct contact with the DNA because of the lack of membranes. The spectrophotometric analysis for the absorbance ratio, 260-80, resulted in an average of about 1.8. This indicates that the extracted DNA was free from protein contamination. In the preceding PCR, the results were clearly recognizable bands, demonstrated the high quality of the isolated DNA.